term dorsalize, I think, might be confusing some students. It just means that it's associated more with the dorsal side. Okay, there are different types of mesoderm, which we'll talk about next lecture, but you can imagine, you can identify anatomically, there's different types of muscle, for example. The muscle on your back is different than the muscle sort of on the side of your ribs, is different than the muscle on your ventral side. There are different muscles. There are muscles on your dorsal side, muscles on your ventral side, muscles on your, on your lateral side, muscles on your ventral side. They're different anatomical muscles. So when you're dorsalizing, you're making things more dorsal. You might take a muscle that's normally found on your side and it'll become a back muscle. It's dorsalized. Okay, it's about its position within the dorsal ventral axis. Perhaps that's almost easier to see in the anterior posterior axis. We'll talk a little bit about anterior posterior axis when we talk about mesoderm and pseudogenesis, maybe this lecture. But you can imagine your endoderm. Right, your endoderm is going to form your gut, amongst other things. It's going to have dorsal ventral aspects, but it's going to have anterior posterior aspects. Right? So you can have things that make things more anterior, things that make things more posterior. Your gut is made up of specific things. You have your mouth at one end, you have your esophagus, you have your stomach, you have your small intestine, you have your large intestine. Those are all sort of going from anterior to posterior. Okay, there are ways to shift things. You can make things more anterior. If you make things more anterior, you might get rid of the large intestine and everything becomes more anterior and there is no large intestine because things are anterior. Same when you make things more dorsal. If you make things more dorsal, make mesoderm more dorsal, there is nothing that's ventral. There is no ventral muscle or ventral blood because you make things dorsal. Okay, so this idea of dorsalizing, it's literally sort of almost like an anatomical thing. You're making something that's anatomically more towards the back. Whether it's skin for the ectoderm, whether it's muscle for the mesoderm, whether it's some components of the endoderm. Okay, it's things that are normally found on the ventral side, you can dorsalize them and make them more towards the dorsal. Right, so kind of zipping ahead, I've been through some of these things. This is all related to maternal determinants. Some maternal determinants do not move with cortical rotation. Some maternal determinants move with cortical rotation. So that there's an overlap of maternal determinants, and then when zygotic transcription begins, these things can turn on other genes and then signaling occurs. So what you're doing here is this band of tissue that's going to become mesoderm. Okay? There isn't necessarily mesoderm present before mid-glandular transition. During mid-glandular transition, these molecules signal to form the mesoderm. Okay? So mesoderm is formed during gastrulation, except for the dorsal organizer. The dorsal organizer is autonomous. It's always mesoderm, it's always dorsal mesoderm, no matter what you do with it. Why? Because it has these determinants of beta-catenins, adipose related, and veg-T or veg-1. Right? So if the embryo is making mesoderm as gastrulation is beginning, and dorsal organizer is special because it's turning on specific genes. One of the main genes is this transcription factor called goosecoid. So goosecoid is making the cells of the dorsal organizer. Those cells are going to become dorsal mesoderm, this thing called the notochord. And then the notochord can dorsalize other things. It can make other things more dorsal. How does it do that? It does so by secreting things. So the cells of the dorsal organizer, which will be the cells of the notochord, are secreting molecules such as noggin, cordon, follistatin. And we're talking about how that works. So noggin, cordon, and follistatin are found in the dorsal organizer. When the dorsal organizer forms the notochord, they are found in the notochord, and they are secreted by the notochord. So those molecules, noggin, cordon, follistatin, are all very similar. Right? They're found in the right place. They're found in the dorsal organizer. They're found in the notochord. If you dorsalize an embryo, if you make the embryo more dorsal by treating with lithium chloride, which makes beta-catenin everywhere, you get more noggin or cordon or follistatin. If you UV treat an embryo so it can't undergo cortical rotation and there is no beta-catenin, you will not have noggin and cordon and follistatin because you've never made a dorsal organizer. There is no goosecoid. Right? If you have a ventralized embryo and you inject noggin or cordon or follistatin, it will partially rescue that axis. It will make another dorsal organizer, or do its best to make a dorsal organizer. They're redundant molecules because forming dorsal is very important. Good overview, hopefully? Alright, so these molecules are all molecules that are found, that are secreted by the dorsal organizer and the notochord. Some of these are sort of in the same family as the TGF-beta molecules. So when you look at things like early experiments people did, they isolated this thing called activin. Activin is a TGF-beta family, superfamily member. It does play a role in making mesoderm. It's not that important. It's another one of these redundant molecules. However, it was one of the first isolated, and people realized you could use actin to induce mesoderm. So you can take ectoderm, add activin, and you can make different types of mesoderm. It's a morphogen. The more you add, you get dorsal mesoderm, or lateral mesoderm, or ventral mesoderm, depending on how much of this molecule you add. So you can do that with a lot of molecules. A lot of TGF-beta molecules do this. So far, so good. Then people got to follistatin. Follistatin, they realized there's another property of these molecules. So whether it's noggin, or cordon, or follistatin, another thing that these molecules do is they inhibit BMP. So yes, they are secreted, yes, they dorsalize other things, but another thing that they do is inhibit BMP. And why is that important? Well, it turns out that BMP is actually signaling from the ventral side. Right? So when you take this animal cap and it's forming epidermis, if you don't treat it, the reason it's doing that is because BMP is signaling. We know on the dorsal side, you have all these molecules in the dorsal organizer, but on the ventral side, you actually have, it's not quite a ventral organizer, but there is high levels of BMP on the ventral side. Right, so on one side, just before gastrulation starts, you have glucoid, on the other side you have BMP. BMP is actually inducing epidermis and inducing ventral. So it's trying to make things ventral, and it's trying to make things epidermal. So ectoderm doesn't necessarily become epidermis, it has to be signaled to become epidermis, and during development that does occur. BMP is inducing ectoderm to become epidermis. Otherwise, ectoderm will become neural, so the ground state, or sort of the basal state of ectoderm is to become neural tissue in vertebrates, in us. Okay, so BMP is needed to signal ectoderm to form epidermis. The dorsal organizer and the notochord are secreting molecules that inhibit BMPs. Right, so noggin and cordon and follistatin all inhibit BMP. So the reason you can form a neural tube, the reason you form the neural structures right next to the notochord is because the notochord is inhibiting BMPs. Okay, so now I'll slow down. Okay, so you have the ectoderm, the outer layer. So this is looking at the dorsal side of the embryo. You have the next layer down is the mesoderm, and then below that will be the endoderm. The mesoderm is going to be subdivided into different things, one of them being the notochord. The notochord is the most dorsal structure. The notochord forms through convergent extension. The notochord elongates, and it becomes a distinct structure, the cord, the notochord is the name. And you will see that this occurs as you go laterally. You'll form things called somites, then you'll form intermediate mesoderm that gives rise to kidneys, then you'll form lateral mesoderm that gives rise to blood and other things. Right, so the mesoderm divides itself up, but the most dorsal mesoderm is the notochord. Everyone understand what we're looking at? So posterior is in your direction, anterior is into the board. This is the notochord on the dorsal side. That's good? People have known for decades that the notochord can induce the ectoderm, or people thought that the notochord induced the ectoderm. So there are signals in the notochord, there's noggin, cord, and follistatin, they're also treated by the notochord, and the ectoderm above folds and it forms the neural tube. So people thought, all right, the notochord is inducing the ectoderm to form neural. But that's not really what's happening. What's happening is the notochord is inhibiting BMP because there's BMP throughout the ectoderm because BMP is signaling from the ventral side. So all of this has BMP in it. It's going to form epidermis. The only place it doesn't form epidermis is right above the notochord. So right dorsal to the notochord, you're inhibiting BMP, right? So the region that BMP is inhibited in, right? So you're inhibiting BMP, that region can become neural. So it's not so much that noggin and cord and follistatin are inducing neural, they're actually inhibiting BMP to allow neural to form. That make sense? And we'll talk a lot more about this when we talk about neurulation. Questions for this so far?
So, sorry, right? It's the region right underneath the secondary region. When the notochord, cordon, and follistatin inhibit BMP, that region folds to form
The neural tube. So this is looking at, this is in situ hybridization, so this is the message for, in this case, noggin. So this is where noggin is transcribed. So noggin is found in the dorsal organizer, then with gastrulation, noggin is found in this dorsal mesoderm. Okay, so this is noggin in the dorsal mesoderm. This is looking from the side of the embryo. This is looking dorsally on the top of the embryo. When you look at where noggin is found in this structure, which will become the notochord. So noggin is found in this structure. That is going to be the notochord. The notochord is the dorsal mesoderm. It has noggin and cordon and follistatin. There's ectoderm above this. Okay, this is the mesoderm that you see. This is the dorsal mesoderm. There's ectoderm above it. You can see it in this diagram, right? So this is the mesoderm, and above it is ectoderm. The notochord is going to secrete noggin and cordon and follistatin. It's going to secrete it to that ectoderm. So this region of ectoderm is going to fold, and you're going to form a neural tube. So the neural tube is going to be right on top of the notochord. So in little diagrams like this, these little arrows are trying to signal to you that this red stuff, this dorsal mesoderm, is signaling to the ectoderm. That ectoderm is then going to fold. So right dorsal to the notochord, you're going to form a neural tube. Okay, and that's because the ectoderm, this neural tube was ectoderm, but that neural tube now has BMP inhibition, and the ectoderm became this neural tube. We'll talk about neurulation, like I said, in a couple of lectures. But it's important to appreciate that the ectoderm is forming neural because BMP is inhibited. Why is BMP inhibited? Because of noggin and cordon and follistatin. Right? So BMP is something called bone morphogenetic protein. There are many BMPs. I think there's 14 or 15 different BMPs. They were first discovered in bone. That's why they're called bone morphogenetic proteins. They do play a role in bone development, but they play lots of roles in other tissues. They just happen to be called BMPs. How do we know that BMP is the epidermal inducer candidate? How do we know it actually makes epidermis? Okay, and you can do experiments to prove that it is the candidate that induces epidermis if you inject excess amount of BMP. So normally, at the beginning of gastrulation, you have goosegoid on one side, BMP on the other side. There isn't BMP on the dorsal side. There isn't a lot of BMP at the dorsal organizer. However, if you inject mRNA and you have BMP everywhere, you will get epidermis everywhere. You will not form a dorsal organizer, and you will not form neural tissue. So if you overexpress BMP, you get no neural. It's all epidermis. So BMP is just going to make epidermis everywhere. Make sense? So it's overwhelming the dorsal signal. There is no dorsal, there is no neural, just epidermis everywhere. The other thing you can do is knock out BMP. BMP works through the receptors, it's TGF-beta signaling. You can make dominant negative forms of the receptor so BMP cannot signal, right? So if you get rid of BMP, there is no BMP signaling. What happens is that the dorsal organizer spreads. There's nothing to inhibit dorsal, right? Dorsal can spread, and eventually you get dorsal tissues that are found on the ventral side. You're kind of dorsalizing the embryo because there's nothing to prevent dorsal from spreading. So if you inhibit BMP by knocking out the receptor, you're going to extend the size of the dorsal organizer. You're going to extend sort of dorsal tissues. This also happens if you inject BMP inhibitors, BMP binding proteins such as follistatin and cordon. Wherever you put follistatin and cordon, you're inhibiting BMP. You're potentially going to make neural, or you're going to make more sort of dorsal-like structures. Does that make sense? How is BMP doing this? So BMP signals and induces ectoderm to become epidermis. How is it doing this? It's doing this by these two other genes, right? So on one side you have a dorsal organizer, the glucoid, and you're going to cause the secretion of cordon, follistatin, and noggin. On the other side, you have BMP. It's going to make things more ventral, and it's going to make things epidermis. It does so by these two genes that it causes the transcription of. One is called MSX1, and MSX1 is actually the gene that makes ectodermal cells epidermal. So MSX1 gene induces epidermal genes. So MSX1 is the downstream candidate from BMP that makes the ectoderm epidermis. There's another gene called VENT1, that makes things more ventral. So this gene is preventing dorsal things from spreading. It's keeping things ventral. So it's a battle between things on one side trying to make tissues dorsal, and BMP on the other side with VENT1 and MX1 making things more ventral. Is that okay? So that kind of summed up in this little diagram here. Okay, you have endoderm, you have ectoderm. At the beginning of gastrulation, you're going to get signaling occurring. You're going to start making mesoderm. Okay, that mesoderm is going to be induced. We'll talk about it hopefully in a few minutes. Okay, this mesoderm is going to be induced, and this mesoderm is going to have different characteristics. On one side, it's going to be more ventral. Why is it more ventral? Because it has ventral molecules such as BMP and VENT1. On the other side, it's going to be more dorsal. Why? Because it has dorsal molecules such as glucoid and noggin and cordon and follistatin. The dorsal organizer is more important because it's autonomous. If you remove it, it's going to form dorsal organizer anywhere. The ventral side does not do that. If you transplant the ventral side, it is not going to form ventral tissues by itself. It needs other signaling molecules, so it's not autonomous. Only the dorsal organizer is. So what do we see here? This is a chick embryo. So you have a areopaca, areopellucida, you have the chick embryo here, the epiblast is here, and you have Henson's node is here. So Henson's node starts at the anterior, and Henson's node is moving towards the posterior. So Henson's node is moving towards the posterior. As it does so, it's going to leave behind it the notochord. So it's making this notochord from anterior to posterior, and the notochord is going to get longer. At this stage, this is where Henson's node is, this is where the notochord is. In this case, this is an in situ hybridization for noggin. So noggin is telling you exactly where the notochord is. So this is where noggin is expressed. If you then look to see where is BMP expressed, BMP is expressed everywhere except relative to this noggin. Why? Because noggin is a BMP inhibitor. So wherever noggin is, BMP isn't.
And downstream of BMP signaling is SMAD1, same idea. You can see SMAD is present in most of the embryo, making it more ventral. This part, where noggin is, can be more dorsal, because you're inhibiting BMP. This little diagram or something like it will be somewhere probably on the final exam or something. So hopefully it makes sense, but let's just do it again to make sure, right? So you can see at the top, you have BMP coming from one side, and from the other, you have noggin coming from the other side, right? What are the sides? Right, so this is going to be, this is sort of the dorsal side, this is the ventral side. Right, so on one side there's high noggin, on the other side there's high BMP. You're gonna put different types of mesoderm in here depending on the overlap of these two. So dorsal mesoderm, the notochord, where is the notochord? The notochord is gonna be present, what color do they don't have that color? I'll just use blue again, I guess. The notochord is gonna be present where there is no BMP. Right, the notochord is inhibiting BMP, so the notochord is gonna be present on the dorsal side. So they're trying to show you that over here somewhere, right? The notochord is gonna be present on the dorsal side where there is no BMP. Make sense? Going the other way, you're gonna form mesoderm on this side that's ventral. So blood is ventral, we'll learn that soon. So blood is gonna be present on the ventral side where there is BMP, but there is no noggin or cord or follistatin. And then you can put different things in here depending on how things are overlapping, right? If you have a little bit of noggin but a lot of BMP, you can put in here things such as kidneys. And we'll see that next lecture, right? Where you have more BMP but a little bit of, more noggin but a little bit of BMP, over here you can put muscle. I don't know why I can't spell muscle. Okay, so you have different tissues, notochord, that's totally dorsal. Next you're gonna have muscle, which is a little bit more ventral. Next you're gonna have kidney, which is a little bit more ventral. Next you're gonna have blood, which is a little more ventral. And it's all related.